indicated on Monday, we'll go back through and review implantation, all right? And then we'll use those same images in your lecture notes to look at the changes in the formation of the embryonic disc and the trilaminar disc and the folding and the formation of the 3 basic germ layers, all right? And then that's where we stop. And we have 2 things for lab. We have the quiz on female reproductive, and we have the sea urchin fertilization, which the Thursday wasn't scheduled to do until tomorrow night, but we went ahead and did it last night because the sea urchins came Tuesday morning. And so we made zygotes of baby sea urchins. They didn't really develop into sea urchins. So we'll do that, and then they're still open lab, but your last lab time to study because we have our exam on Monday, all right? So the lab exam will be first because that's timed, and it will be single questions on PowerPoint slides, similar to your quiz. Everybody will have dividers up, everybody will answer the same questions, so it won't be the A, B, C, D format, but you need to bring a Scantron because it will have multiple choice as answers, all right? So instead of identify this organ from endocrine histology with 4 different images. It'll be just 1 image and 4 or 5 answers, multiple choice, and everybody will answer the same question, okay? So I typically have the rows connected across here for the first 2, so people are spread out a little bit more. And then that'll take about an hour and 15 minutes by the time we go back through them once. And then about a 15 to 20 minute break, and we usually start the lecture exam around 7:30, okay? So anyway, that's our setup for Monday. So you'll need 2 Scantrons, 1 for the lab, the long 882 ones, 1 for the lab and 1 for the lecture, okay? The lecture exam is typically around 65 to 70 multiple choice questions for the Scantron, and then about another 30 questions of short answer, fill in the blank. I promised you the table of chromosomal patterns. What's the chromosomal pattern of this particular cell? Another table of homologous, so what's the male structure that's homologous to the labia majora, or what's the female structure that's homologous to the prostate gland, or what does the Golgi apparatus develop into, or the oviduct develops from which embryonic structure, those types of questions, okay? So about 8 or so of those, yeah.
I don't know if you already said this, but do you need to remember like which specific phenomenon is which ones are protein, steroids, stuff like that?
For the lecture exam, if I give you a molecular like cholesterol, if I show you a diagram of cholesterol with the 4 rings, you should recognize that that's going to develop into a steroid, or the fatty acid, that's going to form the arachidonic ones. So there's a table, or a form of a table where I kind of went through, not the one with all the tiny print, but the next page. There was a table where I gave a sample of the glycoproteins, which were really long, 50 or 75 amino acids in the modified, yeah. So you don't have to memorize a list, but if I show you the molecular structure, what would it be classified as? Yeah. Okay, so I did not show this to you last time. I kind of skipped over the slide that reminded me to do it, but this is called intracytoplasmic sperm injection. It's a form of in vitro fertilization in which for 2 reasons. One, the sperm can't swim, so it's not going to make its way into the ovum, or there's some genetic issue that they want to pick out a specific sperm that may have been modified, or they're doing an X or Y chromosome sperm, and so they will select one specific sperm. So it's taking up a sperm. This is a glass pipette that's been heated and the glass drawn out really, really thin. This is a larger pipette here that's creating a suction to hold the egg in place. You're seeing polar bodies down here, okay? And you can see the sperm go out right there. There's another one, a very similar one that shows they're actually jostling. So you can see how this is creating the suction to come up close. They draw some fluid, some of the cytoplasm out and push it back in. So this is the corona radiata. And still not the one I'm looking for that shows the polar bodies, if I can. There we go, that one's showing. So you can see this structure right there. That's actually the first polar body. They're trying to get it out of the back. So that's the first polar body there. This is the secondary oocyte. So we have a 2 parentheses in and a 2 parentheses in. This would be the corona radiata follicular cells. And so by accident, it was found by puncturing the plasma membrane of the oocyte with the pipette. Whether or not they put a sperm in there will trigger that cell to undergo division and to go from the secondary oocyte to the ootid. This takes a while they're thinking about it, and I'm sure there's talking going on while they're doing it. This is one I showed last night. So this is very similar to our model over here. We have 1 and all right, so we hold it like this. This is kind of what you're seeing here with the secondary oocyte and the first polar body, okay? And this is showing the same cell, but with the sperm penetrating the plasma membrane, okay? There's the little guy there. So ICIS, intracytoplasmic, ICSI rather, intracytoplasmic sperm injection. So this is, so if they take sperm from the head of the epididymis rather than the tail, it's going to have this procedure because it's not going to swim into the sperm, into the oocyte itself. Okay? All right. All right, so which of the following, I'm going to drill this into whether you like it or not.
Let's go.
Which of the following identifies the chromosomal pattern of the second polar body?
2 fragments anyway. Wait, just add? I just thought it was 2 Oh, that's right. There's
Well, I got a warning on my car last night when I left to go home. I had students here until 10:15, and then I went and I ran off exams that I was giving at Sac State. And it said, no visible parking decal of daily ticket. Well, I had my faculty permit in the proper location in the lower right window. Vehicle in violation of parking restrictions. I assume it was saying, I don't know what that was for. And then it says, no overnight parking, because I left at 11:45 p.m. And apparently overnight parking starts at 11 p.m. So it was a courtesy warning. So I'm gonna call and say, did you notice that the faculty parking permit was up there? Do I have to call and ask permission to stay after 11 and work? So I think it was because my car has Colorado plates, so they just didn't bother looking for any other official documentation. But anyway, after 23 years, I got my first parking ticket, not a ticket, a warning. Okay, so we went through all of, any questions about this stuff that we talked about? Because I'm moving ahead to review the implantation, all right, because this is our lecture on Monday night. But quiz on that, question quiz. So what is the event happening over here this is not an oocyte, this is a spermatozoan. So which of these events is being illustrated by this animation?
Yeah. Because remember the membrane was bursted. It was doing the that's where the sperm goes in. Yeah.
Sure.
That's when
Sure.
it ruptures.
The only thing that's different is the follicle. Okay? So I'm going to stop this. Here's a question to review about the follicles and the oocyte stages. So the terms primary and secondary are used both for the egg cell itself, the oocyte, and for the follicle. All right, so two different things are going on. The oocyte is going to be a primary oocyte after the female is born, all the way through until ovulation. So if a oocyte is not ovulated, it's going to be primary, whether it's in a secondary follicle or a primary follicle or even an antral follicle. Even a mature antral follicle is going to contain a primary oocyte until just like 40 minutes before ovulation, okay? When it undergoes its second meiotic division. So whether we're going to be looking at a primary oocyte, meaning it hasn't yet undergone its first meiotic division, that's the oocyte, okay? Then the follicle, if it has flat squamous, a single layer of flat squamous cells around it for the follicular cells, this is a primary follicle with the follicular cells surrounding a primary oocyte. So the oocyte chromosomal pattern is a 2 parentheses 2N. The cells, the follicular cells that are the primary follicle cells are diploid, just a straight 2N, like every other cell in the body that's not one of the gamete cells, okay? Then, still a primary oocyte, but if the follicular cells have rounded up to form a single layer of cuboidal cells, then this is now a secondary follicle, which still contains a primary oocyte, which is still 2 parentheses 2N, and the follicular cells are still 2N. But they started to obtain more of the smooth endoplasmic reticulum and starting to convert the androstenedione from the theca interna cells, which aren't very many at this point, into estrogen. So you're starting to see an increase in the amount of estrogen. All right? Then those follicular cells undergo mitosis and multiply, and we have 2 to 4 or 5 layers of follicular cells. Still a secondary follicle. But we obviously are producing more estrogen because we have more of those follicular granulosa cells making estrogen. So the amount of estrogen is increasing, but still a secondary follicle, okay? And still a primary oocyte. Then we move into the antral stage in which we have a few pools of fluid in between the follicular cells. And that's an antral follicle, okay? It's still an antral follicle, even if those pools coalesce, if they form a small space. So we can see a small space here. It's still an antral follicle, still a primary oocyte. And I just haven't drawn in the zona pellucida, and I haven't drawn in the theca interna cells, which would be visible easily by the time we reach that stage. And then the antral follicle is going to get larger and larger and larger, all right, for the 2 months or so before it actually ovulates and ruptures. And so about 40 minutes to an hour prior to ovulation, our primary oocyte is going to undergo the first meiotic division. And that surge of LH is going to cause an increase in the pressure of the follicle. And we have released a secondary oocyte and our first polar body. We'll go ahead and put the zona pellucida around that. And it's going to be present here too. Okay. So at ovulation is the first time that I ask you to recognize that the oocyte is a secondary oocyte. Okay? And it's going to have the corona radiata and some cumulus oophoris cells. And so obviously this is the mature or Graafian follicle that has ruptured. So at that point, the oocyte is a secondary oocyte. And that and the first polar body both have a chromosomal pattern of 2 parentheses in. Okay? So secondary oocyte first polar body. Those are the products of the first meiotic division. And it's going to stay like that and get flushed out of the female reproductive tract out the vagina unless it gets fertilized by the sperm. When the sperm fuses to the integrin alpha-6 beta-1 protein on the egg membrane and is pulled down into the egg, that triggers the second meiotic division. And so what we have inside that, I'll erase this one over here. So then we have our secondary oocyte with its 2 parentheses in chromosome. We have the first polar body, all right? And here comes the sperm with its N chromosomes. As a result of that, this cell will divide. The whole secondary oocyte will divide unevenly again to create the second polar body, which is N, and that's where half of these chromosomes are going to go, is into that second polar body. So now this is just N, and it's an ootid, also known as an ovo. So we have the female called the female pronucleus, which is N, and the male pronucleus, which is N, that are separate, okay? When they come together to match up as a new 2N, so then they're going to do this. We have the male's chromosome and the female's chromosome merging together into one nucleus that is now a 2N. That is our zygote. A single cell with a unique pairing of 2 N chromosomes. And out here we're still going to have our first, which is 2 parentheses N, and our second, which is just N. But inside the zygote, it's going to be 2N. Okay? Now occasionally that first will divide again, but I'm not going to do that. Okay? Well, it doesn't always do that. I'm not going to have it do that. Does that help?
For the secondary oocyte, what is the name of the follicle?
It's a mature antral follicle that it's leaving. Once it's ovulated, it becomes the corpus hemorrhagicum because of the pterium bleeding as for ovulation, and then it becomes the corpus luteum. and then the corpus albicans once it stops secreting hormones. So we don't call it a follicle anymore after ovulation, it's the corpus, whatever stage. Okay? All right?
I have a question.
Yeah.
So for the Graafian follicle and the antral follicle, are those interchangeable, or is there?
Mature antral and Graafian are interchangeable.
Okay.
An antral is just anytime we start to see those pools of fluid, even when they coalesce, if it's still a small size, and we don't see a very strong cumulus oophrys, that little stalk that it sits on,
Yeah, okay.
okay? Anything else? Yeah.
For review, what enzymes are involved in making that estrogen with the second follicle with those cells?
All right, so starting with the theca interna,
Yes.
under influence of LH, is in the mitochondria, it's gonna take cholesterol and convert it to pregnenolone. And then in the smoothie R, that's gonna be converted to androstenedione. All right, that's under the cyclic AMP activities. And then that will move across the basement membrane into the follicular cells, which are homologous to the Sertoli cells. And their aromatase is gonna convert the androstenedione to estrogens or estradiols. A little bit of that happens in the Sertoli cells, but not nearly as much. All right, so the 5-alpha reductase that forms the dihydrotestosterone, the female doesn't do because she doesn't have male phenotypes. And that's what's being used for the secondary sex characteristics of the male.
Do we have a diagram for that?
It's in the lecture note with female physiology. It's the same events that occur between Leydig cells and Sertoli cells, but in the female it's theca interna cells and follicular cells. All right, so we had it fertilized, moving through the oviduct, and remember it forms this solid mass of cells, which is the morula. And by the time it gets to the uterine cavity, it's formed a blastocyst or blastula. Either term is fine. And the blastula is a shell of a single layer of cells, all right? With a cluster of cells on one end called the inner cell mass. The single layer of cells is known as what? Do you remember?
The
No,
journal?
zona pellucida is still there, but it's not, zona pellucida is not cells, it's a protein coat. Trophoblast, okay? So the zona pellucida, this is called blastula hatching. So the zona pellucida ruptures. Did I show you guys that on Monday? The blastula hatching? See if I- no, I don't think so. There's something wrong with the sizing of the screen here. So we've got the cell undergoing, you can still see some polar body. Here's the zona pellucida, still around. It's undergoing 2-cell, 4-cell, 8-cell, 16, 32, 64, 128, 256. But still captured within the zona pellucida, okay? Before it can actually implant, it has to lose that zona pellucida. That happens in the uterine cavity, or it's known as hatching, kind of ruptures and the blastula squeezes out. So here's the morula with the hollow, the solid ball of cells. You can start to see it starting to hollow out. So we have the trophoblast cells and the inner cell mass. And then just about now, it's going to rupture and you're going to kind of see almost like well, there it goes. Okay? So the zona pellucida ruptured and it's kind of like popping a grape out of the grape skin. And the grape skin would be the zona pellucida, and the grape meat, if you will, inside is the, let's watch that one more time. So this time in the human, it's still in the oviduct, where it forms a solid ball morula. Around day 3 or 4, about around day 4 or 5, it enters the uterine cavity as a blastula. It's starting to hollow in the middle. See the inner cell mass there in the upper right. The zona pellucida is still imprisoning it. But this is the shape without the zona pellucida that actually implants. It's going to rupture here in a second. There. So there's the zona pellucida without it, and now the blastula is free of that encasing protein coat. Okay? That happens in the uterine cavity. And so this is what we have around day 6 or 7 entering the uterine cavity, and then it settles down on the uterine surface around between day 7 and 8. By the end of day 9 to 10, it's settled under the surface of the epithelium, okay? And started the development of the placenta. So here it still has the zona pellucida. They didn't show the hatching, but it lost the zona pellucida, and it settles down with the inner cell mass towards the surface of the uterine lumen. This is the stratum functionalis up here, so it's moving that direction. All right, so let's stop it there. So your diagram kind of shows it sideways, and I'm going to flip it around so it's actually sinking down this direction. All right. May I erase the board? So we use the same images for implantation that we use for embryonic development. We'll look at the formation of the placenta first, and then we'll come back and look at those same images but focusing on the inner cell mass, okay? So what I'm going to draw is actually, thinking it this way, I like this video, but it shows it going up. So here's the uterine lumen. And we have the surface of the uterine cavity. It's going to have some uterine glands. Anybody remember what those secrete? They become white and kind of frothy. And their purpose is to provide nutrients for the implanting tissue, glycogen, which is a bunch of carbohydrates, all right? Now I haven't drawn that very long or very coiled, but remember this is happening as far as our ovarian cycle, our uterine cycle. So day 14 is ovulation. How long is the egg viable for fertilization to occur? 24 hours, all right? So within 24 hours, so by the end of day 15, it has to be fertilized or it can't be fertilized. So then it's going to be implanted around day 7 to 9. So we're talking about day 21, 22 is when this is happening, all right? That means 22 days after the first day of menses. And then we're assuming menses isn't going, I mean, some people will spot throughout their pregnancy, all right? But typically because of the hormone levels due to pregnancy, menses doesn't happen again for a while, especially if the mom is nursing. So uterine gland. And in those 9 days, as it's moving through the oviduct, these are becoming longer and more coiled. We would be in our secretory phase of the endometrium. And the spiral arteries are growing very rapidly because of the progesterone level after ovulation, all right? So the connective tissue epithelial nature of the stratum functionalis is due to the estrogen, which peaks again, but is not as high as it used to be prior to ovulation. And we have an increase in the amount of blood vessels and length of the blood vessels because of the progesterone, which is now our major hormone after ovulation, okay? So the blastocyst settles down, and I'm going to draw it already having moved. It doesn't digest these cells, it kind of oozes its way in between, okay? Well, I'll go ahead and draw it and then erase it just to give you the idea. So we have our blastocyst, which is a single layer of trophoblast cells. What would be their chromosomal pattern? 2N. And this is the same 2N chromosomes that were created by the zygote, okay? So the same 2N chromosomes that we saw in the zygote, one of the ends came from the dad and one came from the mom, all right, in the sperm and the oocyte. Then we have our inner cell mass, which is also the same chromosomes. Although the cells are looking different. So now the cytotrophoplasts multiply, they undergo mitosis, and they fuse together. So we have one large massive cytoplasm with multiple nuclei from all the trophoblast cells that have fused together. And that's what you're seeing here. Okay, so when this is upside down from the way I'm drawing it, so here are the cytotrophoblast now. Instead of trophoblast, once these cells fuse together, these are known, this tissue of fused trophoblast now is known as the syncytial. A syncytium is a fusion of cells. So once the syncytial trophoblast is formed, and notice we still have cytotrophoblasts on their surface, the trophoblast cells, the individual cells, we now call cytotrophoblast. All right? So when it was just a single layer around the blastula, they were trophoblast. Once the syncytial trophoblast is formed, this was one long, large oozing, kind of like a plasmodium, the rest of the single cells are cytotrophoblast. So as this moves deeper into the stratum functionalis, this is our stratum functionalis of the endometrium of the uterus, all right? So to be accurate, I'm going to bring my diagram of the uterine gland a little bit farther down. And so down here is going to be the stratum basalis. The deep anchoring component of the endometrium. Okay. It's the stratum functionalis that's affected by estrogen and progesterone. And it's the stratum functionalis if the woman doesn't get pregnant, that with the drop of progesterone, the last week of the menstrual cycle, that causes the spiral arteries to constrict and the tissue separates. It separates at this junction between stratum functionalis and stratum basalis. And as we'll see, that's the same region of separation when the baby is born, okay? And you have the afterbirth and the placenta and all the other tissues of the surrounding uterus separated at that level. All right, so if you want to take a picture of that, go ahead, because I'm going to bring the implantation tissue deeper down into the stratum functionalis.
So, diffused, I can't say that word.
Syncytial trophoblast.
Trophoblast, it's still too uneven with the fuse?
It's not really, we don't, because it just has multiple nuclei inside it.
Okay.
And each nucleus would be 2 ends, but there's no longer plasma membrane
Right.
separating them.
Okay.
But I don't know how many nuclei are there, so I don't,
It's hard to.
yeah.
Would that be a test question?
For that, I mean, you should know there's multiple nuclei, but there wouldn't be a way of asking for the chromosomal pattern except to be some factor of 2n,
Okay.
some unknown factor.
A question for you.
Uh-huh.
The fused cells you have it spelled differently on the board and in our lecture packet.
The what cells?
The fused cells?
The syncytial trophoblast? It should be S-Y, oh, I left a S-Y-N, syn, oh, there should be another Y there. And sometimes syncytium is spelled with an A instead of an O, syncytial part. All right, so now, notice how much of this,
Oh my God.
so it's completely settled under, but the greatest form of the syncytial trophoblast is towards the stratum basalis aspect of the implantation, okay? So let me bring that farther down. Because the whole purpose of doing this is to provide the developing embryo nourishment, okay? It's going to get that nourishment from the maternal blood vessels. It's not going to get it just floating around in the uterine cavity. It has to get it by direct bathing, if you will, in the blood of the mother. So now we'll bring this farther down, and we'll have the uterine epithelium closes over the top of this tissue that's burrowed within. So we still have our, make a little bit bigger. We still have our cytotrophoblast. And our inner cell mass. And all these cells are the same 2N as our cytotrophoblast. And then the syncytium now starts to form little finger-like branches as it kind of, like a macrophage or like pseudopodia, kind of flows out into the surrounding tissue. Those finger-like branches are longer and branch more often the deeper we are in the stratum functionalis. Because there's just more tissue to expose and more blood vessels done. There's not any blood vessels up here, all right? So these would have the scattered nuclei of all the cells that have fused to form them. And as it expands, it starts to gain access to uterine glands, all right? It starts to find blood vessels, and it actually opens up the blood vessels, and this incision kind of slides down and lines the inside of the blood vessels, so that the maternal blood is in direct contact with the fetal tissue. By fetal tissue, I'm talking the syncytial trophoblast, all right? Now, there are some animals, like horses, where it doesn't invade the maternal blood vessels. And oxygen has to cross the blood vessel wall of the mother and then cross the connective tissue and blood vessel wall of the fetus to actually get into fetal blood. In humans, in primates, in guinea pigs, they have this direct access to the blood. So the only tissue that has to be crossed is the fetal tissue, okay? I don't know why it's that way. It seems more efficient that way, but we have it that way, all right? So now the cytotrophoblasts start to multiply, and they create columns of cytotrophoblasts that move down into the syncytial trophoblast. Not very much up here, but as we're deeper in the stratum functionalis and forming more of these branches, we get a more extensive branching of the formation of the cytotrophoblasts. So the cytotrophoblasts then further develops from just epithelial type cells to connective tissue, so fibroblasts, all right? And blood vessels will form in there. And so the connective tissue blood vessels that form from the cytotrophoblast is known as extra-embryonic. It's not inside the embryo, all right? So it's extra-embryonic. And the term mesoderm is used. That's basically where most of our connective tissue and muscle comes from. So the next main term, we call the combination of syncytial trophoblast and the cytotrophoblast developing into the extra-embryonic mesoderm. All of that now is called chorion. Chorion is still fetal tissue. It has the 2 N chromosomes that were originally formed as the zygote was formed, all right? So anything chorion is going to be fetal tissue. So we continue to see further branching, even in the term placenta. When we have this model here, so we go from this original, here's the syncytium, kind of drawn that way, the way I drew it on the board. So the dark red would be the syncytium, and the yellow is the cytotrophoblast, as it's sinking down into the stratum functionalis. Once it sinks all the way down, don't worry about this white stuff here, but the dark red is the syncytial trophoblast forming branches, kind of the way I've shown there with the blue tissue, the cytotrophoblast and syncytial trophoblast. And then as it further develops, we see this, okay? So this level of branching here is what I'm showing here. And notice it doesn't branch nearly as much, and it's shorter up near the close to the surface where that it's sunk down below, so up close to the uterine lumen, okay? So that's 2 different types of chorion. Where we have the extensive branching, this is known as chorion villi, okay? Where we have less branching, it's known as chorion, and there's different ways of spelling this. You can go L-A-E-B-A-E, or L-E-A, anyway. It means smooth. So we have the branching chorion and we have the smooth chorion, okay? Again, chorion is fetal tissue, so this would be the chorion villi. And this is becoming our true placenta. And this is the chorion levi, okay? So when we actually look at a larger model of part of the placenta at term, just before the baby's born, this would be the surface of the placenta. Well, we'll get to it. It's the amnion. And here are the chorion villi. Okay? And the spaces in here now are filled with maternal blood. So it's as if you took a new plant that you were getting rooted, in a greenhouse before you put it out in the garden, and you took it out of the dirt and shook it off and looked at it in the microscope, you would see lots of fine little branches and even almost hair-like structures, increasing the surface area. And so that's what it looks like under a microscope, a dissecting scope, like 10x. If you were to, so when I worked on my doctorate, I would come down, I used cancer cells, placental cancer cells, called chorion carcinoma, that I could keep alive forever, okay? And I was studying the internalization of low-density lipoproteins in transferrin, which carries iron. But I did some experiments on actual cytotrophoblast. And I would go to Sutter Memorial, which is torn down now, and for a scheduled C-section. And I would get the placenta, take it back to my lab in a clean, unused ice cream, 2-gallon ice cream container. And with personal protection, goggles and all that, I would chop it up in little pieces because we still, even at term, have cytotrophoblast cells. And through a series of washings, I would isolate those and I could keep them alive for about 7 to 9 days and do experiments on them, okay? So it really looks like little rootlets of a plant that you pulled out of the dirt. And it has all these branches that provide the surface area for nutrients and waste material to be exchanged with maternal blood. Now, you'll notice that we have the maternal tissue down here. Here are spiral arteries, okay, that are supplying that. So the stratum functionalis, not the basalis, the stratum functionalis undergoes an inflammatory type reaction. So all of this tissue here has lots of fibroblasts, okay? They become swollen. There's actually, even though it's an inflammatory reaction, there's a suppression of the immune system. Because, anybody know why? Why would we want to suppress the immune system in the uterus? Half of these cells have chromosomes that didn't come from the mother. So they're making foreign proteins. And in fact, some women have difficulty or cannot get pregnant because they make antibodies against those different proteins from the dad, okay? So there are many cases where a woman has gotten cancer because of her suppressed immune system due to the fact of being pregnant, okay? And unfortunately sometimes has to make a choice between her life and the baby's life, okay? So when those changes occur in the connective tissue out here, it's called decidua, a decidual reaction. So anything that's decidua is the mother's tissue. It's regions of the stratum functionalis, all right? So the decidua up here, associated with the chorion levy, is identified as the decidua capsularis. It's forming a cap over the developing fetal tissue. So this is decidua capsularis. All the connective tissue associated with the chorion levy. The decidua down here, associated with the chorion villi, and still part of the stratum functionalis, is called the decidua, and it's a confusing term, basalis. But it's not part of the stratum basalis, okay? It's the part of the stratum functionalis associated with the chorion villi. Then, all the rest of the stratum functionalis, whether it's on either side of the implanted tissue or on the opposite side of the uterine cavity, is known as decidua parietalis. So I'll show you an image of that. Because obviously as our embryo is growing, it's bulging out this surface of the uterus, okay, and pushing it to the opposite side of the uterine cavity. So here we see it's moving in even deeper, incorporating uterine glands, incorporating some blood vessels. this is showing, I'm gonna stop there because it's showing the development of the inner cell mass, which is not what we're doing right now. So you have this diagram in your lecture notes, all right? So we've already gone past this spot. It's farther down. Notice how it's still covered up by the simple columnar epithelium. You don't have to know all the different chemicals and stuff here. This would be our uterine lumen, okay? But it's submerged below the surface of that uterine lumen. So before we move on to the completion of implantation, so based on that video that we saw a little bit ago, identify the cells in that image.
Because it has the module of the module, right? It has like four mass.
What would the chromosomal pattern be? Do it. Good. We're all done with 2 parentheses, anything. So is this before implantation or after? Before, because there's no other tissue. If you look at that photograph, there's no other cells surrounding it. So in the video, this is what we saw tumbling through the uterine cavity. Okay, so this is just free. There's no cells or tissue around it. So this hasn't implanted yet. Okay, anybody want more time on this question? All right, so now it's completely deep to the surface of the uterine lumen, but we don't have any differentiation between chorion villi and chorion levi yet, okay? We just have the syncytial tissue. Has anybody ever heard of chorionic villi sampling as a test for fetal health? So an amniocentesis is done. So I was 38 when I got pregnant with my twins, who turned 23 on Friday. My boy and girl, my younger boy and my only girl, and we didn't know they were twins. In fact, my pregnancy test was so strong, when I went in to see the doctor the first time, their brother was 5, I said to my OBGYN, please tell me they're not twins. And so it was like 7-8 weeks. He just kind of did a vaginal ultrasound and goes, no, I just get 1 heartbeat. He did one more, maybe a couple more, when I was 12 weeks pregnant. My husband and I and my dad went to Burma and to China. I was born in Burma and my dad was born in China. So we were kind of going back to our old stomping grounds, if you will. Dragging my husband along. When we came back, I was 14 weeks pregnant and I was scheduled for an amnio just because I was going to be almost 39 when they were when they were born, and my doctor wanted to have that done. So I went in there with my husband, and the tech says, congratulations, you guys are having twins. And my husband literally came up out of his chair. And so that meant I had to get stuck twice. So you have to put in a needle into the amniotic fluid for one baby and fetus and one to the other, and they put a little dye to know that they were in a different amniotic sac each time. And so an amnio takes fluid from this space right here, and cells from the baby's mouth or the baby's skin, the fetus has sloughed off and are floating around in that. And that's where they get the chromosomes from to do a chromosomal check. Well, you can see this is a pretty small space, okay? Even at 8 or 9 weeks, it would be a high probability of damaging the developing embryo to go into that space. And so, but if there's concern of genetic disorders, all right, then they can get, this is still fetal tissue, right? It's still going to have the same chromosomes that are in the developing embryo. So chorionic villi sampling can go in around 8 weeks and get some of this tissue, some of the chorionic villi, and not have to risk going into the amniotic space and killing the developing embryo. So that's why chorionic villi sampling is done. All right, so there's the extra-embryonic mesoderm that's going to come down in here and form the core. And so that, along with the syncytial trophoblast, is our fetal. So anytime you see chorion, I want you to think fetal, okay? And anytime you see decidua, I want you to think maternal. So this is actually a term villa. So this is one of the tiny, tiny, tiny little branches. This multiplies over and over and divides again and again and again, all right? So what we see is this pink out here is the syncytial trophoblast. These dark cells are cytotrophoblasts. So even near term, they haven't all fused into the syncytium, okay? All of this area in here is an extra-embryonic mesoderm that has formed connective tissue with fibroblast, and here are fetal capillaries that are branched out from the major blood vessels in the placenta itself. And so this area out here is where maternal blood is. So there's no maternal cell separating the blood from, but it has to cross the syncytium, it has to cross the cytotrophoblast through this connective tissue to actually get into the fetal blood vessels and for oxygen to enter the fetal red blood cells, okay? But that's what you're looking at as the fine continuous branching. Now even here, the chorion looks evenly distributed. So this is just the development of the chorion. So now you can see very few villi here, just small little nubs. So this is the chorion levy, all right, just under the surface of the uterine component, and down here we have the chorion villi. So for our decidual tissue, this would be decidua capsularis, just between the columnar cells of the endometrium. This would be the uterine lumen here. So we have the columnar cells of the endometrium, a little bit of connective tissue, that's the decidua capsularis, and then the chorion levy, all right? Down here, we have the chorion villi, and this would be the decidua basalis. That portion of the stratum functionalis that's deep to it, where the spiral arteries are supplying blood to the embryo. All right, so we have then 3 different deciduas, and we have 2 different chorions. So notice, first of all, how the growing embryo has bulged out into the uterine cavity. So this would be the endometrium with its simple columnar cells continuing around here. This tissue used to be nice and flat against the surface, all right? But as the embryo grows, it causes that surface to bulge out, almost completely obliterating the lumen, never actually fuses together, as we reach term, okay? So this would be stratum functionalis that we're seeing here. Here with this yellow would be the the chorion levy. The branching down here would be the chorion villi. The light lavender here would be the decidua capsularis. This would be the decidua basalis. And all of this would be the decidua parietalis. Okay? So, maternal tissue of the stratum functionalis is identified as which of the following? All right, baby's gone. Umbilical cord has been cut. All right, so after break, when we come back and talk about embryo development, we'll talk about the formation of the amnion. That's going to form from this orange layer here, okay? So this upper part of the orange layer is going to form the amnion, and this lower part is going to form the embryo. The green is going to form our yolk sac. So here is the amnion filling in this space. Here is the chorionic villi that I showed you on the model, which would make this the decidua basalis. Okay, so right along here, decidua basalis. Then we have the chorion levi right here, the decidua capsularis, a little bit of uterine lumen left, and all of this would be the decidua parietalis. Okay? So, sometimes when a woman's labor is not progressing far along and her membranes haven't torn, it's called the water breaking, it's really the membranes torn, the OBGYN will go up through the cervical canal with a crochet hook-like instrument and will actually tear through these membranes here for the water, the amniotic fluid to rush out, and then that usually starts contractions, okay? If it doesn't, they'll start Pitocin. Because typically, especially if the woman is tested positive for staph or strep within she needs to be in labor within 24 hours because it increases the risk of infection. So, what is it that's being ruptured? Not the stratum parietalis, okay, but, I mean not decidua parietalis, but decidua capsularis, chorion levi, and the amnion membrane itself, okay? Now sometimes there is a condition known as placenta previa, in which instead of being on the posterior uterine wall, the placenta, which is usually around 7, this makes it look huge, it's usually around 7 inches in diameter. Anybody know what placenta means? Cake. And there are some people in some cultures that will eat it after birth of the baby, usually cook it, all right? Lots of animals will eat it afterwards. But if the placenta, instead of developing here, develops closer down here, it can even develop across the internal os. And then you have separation, placenta abruptio, where then there can be loss of life, especially of the fetus, as blood is lost and it loses oxygen. All right, so here's what I showed you earlier with syncytial trophoblast, cytotrophoblast, the extraembryonic mesoderm forming the connective tissue, and blood vessels. Okay, so that's what the oxygen has to cross through. So here's the image of the placenta. So once the baby is born, all right, we have the afterbirth, and the next stage of delivery. And this is identical, except that we have a baby there. This is identical to what happens at menses. Remember, the spiral vessels constrict, and there's a separation between stratum functionalis and stratum basalis. But now the separation is between the decidua, which is what our stratum functionalis has become, and the stratum basalis. So all of the decidua, whether it's capsularis or basalis, is going to separate from the stratum basalis, okay? So all of this separates as the afterbirth that is delivered through the vagina, unless it's a C-section, after the baby has been removed from the uterus. Yes?
For a C-section, do they just scrape?
Yeah, I mean, the uterus is contracting and separating from it, so they'll allow that to happen. But sometimes, just like after menses, it doesn't completely separate and there's pieces of it left, and then they have to go in and scrape it, because what happens is the smooth muscle of the uterus constricts, and that helps to decrease the bleeding. So right after, the few days after delivery, if you're a nurse taking care of a postpartum after delivery mom, you want, one of the things you do when you go in and check is to squeeze, the uterus is still pretty high, and you squeeze that because if it's boggy, if it's mushy, then it's not contracting hard enough to keep the blood vessels from continuing to bleed. And so oxytocin, released with the baby's nursing, all right, is still going to go to the uterus and cause it to contract. For some reason, it's typically painful with the first baby, but with the second baby, when the baby nurses, those contractions can be painful, okay? Maybe they just don't want the first time mom to not nurse. I don't know what the reasoning is, but that bogginess is a sign that the uterus is not staying contracted. So the last weekend I worked as a labor and delivery nurse before I came out to California to start my doctorate was not a pleasant one. I had a couple who had been trying for 10 years to get pregnant, and they finally did. And on the due date of the baby, they had a doctor's appointment, and the doctor couldn't find a heartbeat. So she sent the couple over to our hospital and hooked up to a fetal monitor, and we couldn't find a heartbeat either. The baby had died in utero, right at term. And so the OB and the parents wanted to do a C-section. Baby was dead. The C-section is major surgery. And so the other OBGYNs said the baby's already died, the mother's health is not in danger, you don't really have a medical reason to do a C-section. So they gave her prostaglandins to cause the uterus to start to contract and expel the baby, and that caused her temperature to spike up to like 105. And so they put her on a cooling blanket, and it took 3 days for her uterus to get into labor, and another nurse and I delivered the baby. Sorry, that was a long time ago. It was a perfect little boy. No cord around the neck, nothing wrong. So I don't know what had happened. The father, I stayed with the father in the labor room where she had delivered, and the OB took her back into OR to deliver the placenta, and they were gone for almost 5 hours because she started to bleed, because all of this tissue hadn't completely separated. And I usually tell this story when I do the cardiovascular, when I talk about blood clotting, because what she had developed was disseminated intravascular clotting disorder, DIC. It could have been, the baby didn't smell, even though it had been in there dead for at least 3 days. It could have been the prostaglandins that they were giving her to get the contractions going. But it caused tiny, tiny, tiny little microscopic clots to be formed throughout all her blood vessels. And then when she needed the blood to clot, after the decidual tissue was separating, she didn't have any clotting factors left. So they had to give her tons of blood and tons of extra plasma clotting proteins and stuff. So I went to visit them. They were on another floor, obviously, and they were going to try to adopt. It was just too much for all that. But I wanted you to see that relationship between regular menses, regular separation of stratum basalis and stratum functionalis, and what happens between the decidua, which is stratum functionalis, and stratum basalis. Okay, so at birth, we see that same separation that we see every menses between the stratum functionalis and the stratum basalis occurs between the decidua, all the deciduas, and stratum basalis. Okay? All right, so let's look at a couple of pictures of placentas. So when we come back and talk about the embryonic development and the development of the amniotic membrane, so this is the surface facing the amniotic fluid. The baby can reach out and touch that, okay? It's in that amniotic sac. This is the maternal surface. So when you look at it, it has this bumpy nature. So what am I saying when I say this is the maternal surface? What's our maternal tissue called? Decidua. So this is the bottom of the decidua basalis, where it's separated from the stratum basalis. You don't have to know the term, but they're called cotyledons. They kind of look like cauliflower, okay? And so this was where the maternal blood was moving up into all these spaces and gaining access to the chorionic villi. Okay, so this bumpy surface that you see is going to be that area. Let's go back to this image. It's going to be that area right along here. So it's separating. This is going with the placenta, and it's separating between decidua basalis and stratum basalis. Okay. This happens to be twins here with 2 umbilical cords. So this is the deep surface of the stratum decidua functionalis, meaning decidua basalis. Umbilical cord we'll talk about, again, a little easier to see. So I said it's about 7 to 8 inches in diameter.
How much does it weigh usually? This looks really
I'd say a couple pounds, that much, maybe 1.5. When my mom was working OB/GYN, so about the time I started, she worked for about 5 more years after I started working as an RN, they wouldn't even ask the parents for permission to take the tissues. They'd just throw it in a big chest freezer and sell it to industries, make shampoo out of it. Yeah, you can buy a shampoo called placenta. And just use it's high in protein, and they just use it for different products. Now, what happens is usually parents sign up for cord blood, which has stem cells in case their child develops a need later on for some kind of stem cell therapy. Supposedly this is the heart-shaped placenta of a little girl born on Valentine's Day, but I kind of think it was manipulated. All right, so let's take a break, and when we come back, we will come back to the inner cell mass, look at those same images, but focus on the formation of the embryo and the amnion, okay?